Welcome back, my dear students. Welcome back to the channel, and today we are going to discuss fourth segment of the human nutrition. Already 3 segments are on air and you might be benefiting with those 3 segments of human nutrition. And I repeat that human nutrition, this lecture is specifically designed for the students of Cambridge, IGCSE and GCE O levels, but the students taking GCSE exams through other boards like Edexcel, AQA and OCR, they can also benefit from this lecture. And today we are going to discuss about the small intestine, its role in digestion, absorption, and then we will be discussing about the role of liver, pancreas in support of digestion process and role of large intestine. So let's continue with small intestine. Small intestine is the muscular tube that has approximate length 6 meters, and this muscular tube receives acidic chyme from the stomach. At pyloric sphincter, stomach continues to small intestine, and at each peristaltic contraction wave, a little portion of acidic chyme is introduced in small intestine. Small intestine is a lengthy tube of about 6 meters length, but it is divided into 3 segments: duodenum, jejunum and ileum, and all the 3 segments are playing their significant role in the process of digestion and absorption. Small intestine is very important in the digestive system because it is the only organ in the digestive system where all the food grades are not only digested, but also the digested substances are absorbed into the bloodstream and transported across the body. Duodenum receives acidic chyme. This acidic chyme contains HCl, and this is possible that this HCl is likely to denature all the enzymes that are to digest food in the small intestine. Why? Because in small intestine, enzymes need alkaline environment, but the chyme contains HCl that is acidic, so it would likely denature the enzymes. So no digestion is likely to take place in small intestine. So this is naturally prevented by the secretions coming from liver. Liver produces bile that is stored in the gallbladder, and when the digestive activity starts in the duodenum right after receiving chyme, so a little bile is introduced through bile duct into the duodenum part of the small intestine. This bile contains minerals, pigments, but does not contain any enzyme. It does not take part in digestion of food directly, but it creates the environment that makes the food digestion completed in the shortest time. The minerals present in the bile, they neutralize the acid present in the chyme. These minerals also help in defining the alkaline pH in the small intestine cavity. This bile also activates enzymes that are coming from pancreas, and this bile also emulsifies fats. The fats, they are emulsified from larger fat droplets, from larger fat globules to smaller fat droplets or globules. This increases their surface area and it helps the lipase enzyme to digest lipids, digest fats in the shortest time. Now the duodenum also receives secretions from the pancreas through the pancreatic duct. Pancreas produces 3 important enzymes. One is lipase, second is amylase, and the third is trypsin. All the 3 enzymes are produced in an inactive form and they are released as pancreatic juice into small intestine through pancreatic duct. This pancreatic juice has got alkaline pH because all the 3 enzymes are to work in the alkaline environment, and the same alkaline pH around 8 or 9. In some of the books it's written 8.5. In some of the books authors have demonstrated it as pH 9, but both are equally effective pH. So these enzymes have to work in the small intestine. These enzymes are produced in an inactive form like trypsin. It is produced in an inactive form known as trypsinogen. When this trypsinogen reaches the small intestine and interacts with the bile, all already pre-activated enzymes, it gets activated to trypsin, and it is likely to digest proteins into dipeptides and amino acids. Similarly, prolipase is the inactive form of enzyme. When it gets encountered with the bile in the small intestine, it gets activated into lipase and it digests emulsified fats into fatty acids and glycerol. At the same time, amylase that is produced as proamylase, it gets activated when it interacts with the bile in small intestine, and this amylase digests remaining starch into maltose. But remember one thing, these are not the enzymes only added up by the pancreas, but intestine does contain some glands. These are known as intestinal glands, and they produce a secretion known as intestinal juice. This intestinal juice does contain mucus that makes a protective coating around the inner wall of the small intestine. At the same time, this mucus further adds liquidity to the food that is being processed in the small intestine. Along with the mucus, these intestinal glands produce some enzymes, like they produce an enzyme known as maltase that digests maltose, a disaccharide, into glucose. Another enzyme, sucrase, that breaks down sucrose molecules into fructose and glucose. And another enzyme, lactase, that breaks down lactose present in the milk into glucose and galactose. Remember one thing, lactose, maltose and sucrose, these are disaccharides and these are not absorbed into the cells. So they are to be broken down into their simpler monosaccharide units, and that is done by the intestinal enzymes that I have named earlier. Similarly, another enzyme that is produced by intestinal gland, that is known as dipeptidase enzyme, and this dipeptidase enzyme breaks down dipeptide molecules into amino acids. Dipeptide molecules are shorter proteins consisting of only 2 amino acids. So this dipeptidase enzyme breaks down this dipeptide molecule into amino acids. Now in the small intestine, we have got absorbable products, and these products include glucose, fructose, galactose. Fructose and galactose are isomers of glucose. So that's why we will mainly discuss glucose. So glucose, fatty acids, glycerol and amino acids are the digested products of carbohydrates, fats and proteins that are to be absorbed into the bloodstream and circulated into the body. But along with these substances, certain minerals, water and some vitamins are to be absorbed in the small intestine as well. Small intestine has got 3 segments, as I told you earlier. Duodenum is the first few inches of the small intestine where digestion process starts, and that reaches at its peak in the jejunum part where digestion and absorption reaches to the peak. In the last part, absorption of the digested substances along with some minerals, some water and some vitamins, particularly vitamin B12, they are absorbed into the bloodstream. In order to absorb these substances efficiently, inner wall of the small intestine is projected into finger-like outgrowths. These finger-like outgrowths are known as villi as plural, and a single projection is known as villus. These villi are collectively increasing the surface area of the inner wall of small intestine in order to increase the absorption. When we discuss the detailed structure of a villus, we come to know the cells that are lining the villus, they have got their membranes projected into finger-like outgrowths. And these finger-like outgrowths of the cell membranes of the cells that are making the villus, these are known as microvilli. So what is the difference between villus and microvillus, or villi or microvilli? Villi are the finger-like projections of the inner wall of the small intestine. Villi are the finger-like outgrowths or projections of the inner epithelium of the small intestine. Whereas the cells that are making the inner epithelium of small intestine, they also have one of their membranes facing the cavity of small intestine projected into finger-like outgrowths. So finger-like outgrowths of the cell membranes of these cells, these are known as microvilli, and the finger-like outgrowths of the tissue, that is the inner epithelium, this is known as villi. Both these villi and microvilli increase the surface area for absorption. Now, when we look into the villus, what we see, we have got a transport network in the villus. And this transport network includes centrally located lacteal and surrounded by capillaries network. So water, minerals, amino acids and glucose, when they are absorbed by the cells, they are transported into the capillaries, into the blood present in the capillaries, and later they are carried to the liver through hepatic portal vein. As fatty acids are water insoluble, so they are given to the lacteals after absorption, and through lacteals they are transported. Within small intestine, it's not only the digested food is absorbed, but water and maximum water reabsorption is done. Water absorption is done in the small intestine, along with some minerals, particularly sodium ions, and some vitamins are also absorbed in the small intestine. And rest of the food that is not absorbed is known as residue. Why the rest of the food that is not absorbed here? Because that food is not in the form that could be digested, or we do not produce any enzyme to digest that food. For example, cellulose fibers that are present in the plant products. We do not produce cellulase enzyme that could digest that cellulose fiber. So that remains in the residue. Remember one thing, digestion process completes in the small intestine, and absorption of the digested substances also completes in the small intestine. If any of the molecule that remains unabsorbed, that is pushed forward into the large intestine, because during the process of digestion, peristaltic waves also continue to occur throughout the length of small intestine. So gradually, the moment food is being processed, food is being digested, it is also being pushed in the forward direction towards the large intestine. So each peristaltic wave pushes a little part of the food in the forward direction, and ultimately that reaches the large intestine. Before going to discuss about large intestine, I must tell you about liver. All the digested substances, they are carried with the help of hepatic portal vein to the liver, where liver decides the concentrations to be flowing within the bloodstream. That we will discuss later when we will discuss the role of liver in the human body. Now continue with the residue that has been pushed forward into the large intestine. Large intestine has got two segments, colon and cecum. In large intestine, there is no food digestion and there is no absorption of the digested food. That's why large intestine's inner wall is smooth. It is not projected into villi or microvilli, just like in small intestine. Its inner wall is smooth, and that is only meant for the absorption of water and minerals from the residue. At the end of large intestine, residue reaches the rectum, and it contains only the materials that are unabsorbable, that cannot be absorbed at all. So these materials are now taken as feces. They are temporarily stored in the rectum, and when rectum is filled up with the feces, so muscular contraction in the rectum wall will make them expelled out of the anus, what we call egestion. So egestion is the last process of this digestive system, where undigested food residue is pushed out of the body, expelled out of the body, out of the digestive system. One must not misconcept this egestion with excretion. Excretion is the removal of metabolic waste from the body, whereas egestion is the removal of undigested food residues out of the body, out of the digestive system. Now we come back to the liver, and we need to discuss the role of liver, that what particular role does liver perform within the body of the human. Liver is a very vital organ that performs a number of jobs simultaneously. One thing, in sport of digestion, what it does? It produces bile. Bile is extracted from bilirubin that is produced during red blood cells hemoglobin metabolism, and this bile is temporarily stored in the gallbladder, and when it is needed, this bile is introduced in the small intestine, in the duodenum part of the small intestine, in support of digestion, in support of activating enzymes, in support of maintaining alkaline pH in the small intestine. Similarly, this liver decides the concentration of glucose and the fate of amino acids that need to travel the bloodstream. In a healthy person, the blood sugar level must be in the range 80 to 120 milligram per deciliter. And this blood sugar level must be maintained. It must not increase. But through the digested food, lot of glucose enters the bloodstream. So liver takes up the excess glucose in response to the hormone insulin and converts it into glycogen and stores it. So that blood glucose concentration must be kept in the normal range, in the optimum range. Similarly, when blood glucose concentration starts declining, so at the same time, pancreas produces a hormone known as glucagon that interacts with the liver cells, and the liver, in response to that glucagon, digests, hydrolyzes stored glycogen into glucose and releases into bloodstream. So this is another job of the liver, that it helps maintaining glucose concentration in the blood. When blood glucose concentration is high, more than the optimum range, so liver takes up the excess glucose, converts it into glycogen and stores it. And when blood glucose concentration drops below normal, so this liver hydrolyzes the stored glycogen and releases into bloodstream in order to restore the blood glucose concentration and help the body cells to continuously respire. At the same time, this liver also oxidizes the lactic acid. The lactic acid produced during anaerobic respiration in our muscles, that lactic acid is carried to the liver, where liver cells oxidize this lactic acid and reduce the possible toxicity chances. Similarly, the ones who keep drinking alcohol, alcoholics. So alcohol, that is the ethanol, is also absorbed into the bloodstream through small intestine, and through hepatic portal vein it is carried to the liver, and liver oxidizes that alcohol to kill its toxic effects. So alcohol toxicity is also reduced, also minimized by the role of liver. At the same time, certain drugs that could be the part of the medicine we normally take to cure certain illnesses, or certain other drugs that could be taken as a result of addiction. So these drugs are also detoxified by the liver. So liver is playing multiple jobs simultaneously, means it is supporting the digestive process by producing and introducing bile into small intestine. It is helping in maintaining the glucose concentration with the help of hormones, insulin and glucagon. It also carries a process of oxidizing alcohol and lactic acid. It also detoxifies drugs, and urea is also produced within the liver during the process of deamination. Unwanted amino acids, they are broken down during deamination process, resulting the formation of ammonia and keto acids. Ammonia is highly toxic and it needs a lot of water to expel it, to dilute it, to reduce its toxicity. As we do not have that plenty of water, so ammonia is immediately reacted with carbon dioxide to form urea. Urea is less toxic than ammonia, can be retained in the body for some time, and it needs less water to be excreted. So urea is produced by the liver during deamination process and is later carried to the kidney, where it is filtered out and excreted out of the body as a component of urine. Whereas keto acids, these are important organic components. If there is an immediate need of glucose, keto acids are immediately converted to glucose and that glucose is respired. But if there is no immediate need of glucose, these keto acids are converted into glycogen, and when needed, that stored glycogen can be converted to glucose and used. But if liver is already having pre-stored glycogen as a healthy adult liver can store around 100 gram of glycogen, and it is already pre-saturated with the glycogen. So no more glycogen can be stored. So these keto acids are converted into fats. Some of the fats are stored in the liver, and some of the fats are transported to the adipose tissues where they are stored in the fat cells. So this is the job of the liver. This is another job of the liver. So in this fourth segment of human nutrition, we have discussed the role of small intestine in the process of digestion and absorption. We have also discussed the role of liver and pancreas in support of digestive activity in small intestine. We have discussed the role of large intestine, and we have also discussed the multiple roles performed by the liver. And remember, assimilation is actually the utility of these absorbed substances into the bloodstream, just like we have discussed about liver, that it takes up glucose and converts into glycogen. Similarly, body cells, they take up glucose for their respiratory needs, they take up amino acids to make their required proteins, and mineral ions for their needs, and fat molecules, fatty acid, they take up to make their membranes. So this is what we call assimilation. So with this discussion, we have ended up with the fourth episode of human nutrition, and with this we have also completed the human nutrition discussion as well. So stay tuned for upcoming topics. Thank you very much.